So please welcome our first guest speaker on the topic, Scott Saleska. Scott, you are a professor of ecology and evolutionary biology at the University of Arizona, where you research biology as part of the global climate system. Your studies focus on how individual or community-level ecological and evolutionary mechanisms propagate to ecosystems and regions and affect large-scale climates. Today, Scott, you are presenting us acquiring data to scale from molecules to ecosystem. So the floor is yours.
Thank you so much. Again, good morning. Thank you for that introduction. I'm going to kick off today's session on data acquisition by talking not so much about individual sensors, but about technologies that are and will enable us to tackle an exciting and a fundamental grand challenge of biology and Earth system science, which is how do we scale from molecules to ecosystems? And of course, in order to do that, we need some key enabling technologies. And I'm going to acknowledge our forebears in this. Isaac Newton once said, "If I have seen further, it is by standing on the shoulders of giants." Well, if Newton can do it, we can certainly acknowledge our giants on whose shoulders we stand, and I will do that in a few slides. So let me get started by showing how we do this in a couple of example systems. The photos here are the two example systems I will talk about. On the left is a thawing permafrost mire in Arctic Sweden, which of course is a key sentinel ecosystem for understanding feedback to climate change. And second, on the right, we have our amazing and unique tool for advancing scaling science, University of Arizona's Biosphere 2. So with that, let me go to the first slide and address the question of what does it mean to scale from molecules to ecosystems? This diagram depicts microbial life. And according to what we sometimes call the central dogma of biology, scaling really just is information transfer. And we go from the DNA, which encodes the metabolic functions of the cell, to transcription of RNA for the functions needed at a given moment, to the translation to proteins, which of course catalyze the reactions that make life go. Next slide. Then the cells, of course, are multiple cells in populations, and the next slide or two show that those populations are part of communities, which all aggregate those metabolisms of the individual cell to produce outputs, which go to the next slide, interact with the environment, next slide, and show ecosystem outputs at the Earth scale. And of course, these include things like gases like carbon dioxide and methane, which are part of the Earth's climate system. And as we know, next slide, the Earth's climate is changing. So we see here that in order to understand climate change at the global scale, we really need to tackle this challenge of scaling all the way back down to the metabolisms that generate the carbon gases that regulate climate. So go on to the next slide, and we can see that I'm going to talk about three challenges for acquiring data to do this, to address this challenge. So next slide, we have first fundamentally acquiring and most importantly, understanding the molecular scale data which we get from these technologies of metaomics. To secondly, acquiring linked ecosystem biogeochemical data at the ecosystem scale. And it has to be linked in the sense that it has to be able to go back to that step number one. And once we have these relationships or observe them in the environment, we really want to be able to test them. And so that's the third challenge, is designing ecosystem scale experiments to look at this. So let's go to the next slide. And we will see that I'm going to talk about this example of methane and carbon cycle responses to permafrost thaw in the field through this project called the Isogenie Project. Our site is a mire in Arctic Sweden. The study integrates ecosystem scale methane fluxes and their isotopic compositions with the dynamics of the microbes that produce that methane. And in order to do that, we need the tool of metagenomics. And finally, I'll talk about challenge 3 at Biosphere 2 when we get there. So next slide. This project is the Isogenie Project, which is a collaboration that I started over 10 years ago now with my then postdoc, Virginia Rich, now colleague at professor at Ohio State University, and a fairly large international team because we're integrating multiple technologies from different disciplines across these scales. So if you go to the next slide, we can see, I can tell the story about what we've done with this at the mire. Here's the mire seen from a helicopter. Next slide. And then we can see that the next slide. Here's sort sorry, back up one. This is the different ecosystems across the mire that's thawing permafrost. We go from dry permafrost pulses to sphagnum-dominated bogs, which are partially thawed, but not completely thawed as it sinks into the water table of the system. And then finally, fully thawed fens, which are completely inundated. And this is a chronosequence that we can see, observe the changes that happen with the thaw. Go ahead to the next slide now. And the challenge, of course, we want to look at global climate change and feedbacks to that. But in order to do that, we need to do this scaling problem. And here's a simple first order test, which is just simply, can we look at molecular data across this thaw gradient and look at biogeochemical data at the ecosystem scale? And can we see a concordance? So that's what I'm going to just show that we can do first. Next slide. The challenge here is to measure those things, the genomic data and the biogeochemical data. And here, this slide just shows that how the technology has really advanced from the days when we had the first draft human genome back in around 2001, where there was over $10 million to sequence a human-sized genome. And now we're down to less than $1,000 for a human-sized genome, which means we can now sequence whole ecosystems and all the genomes of all the microbes of that entire mire and other places with these technologies. Next slide. And what my group brings to this, as shown on the next slide, is the acquiring ecosystem biogeochemical data. And what this is a laser spectrometer, which can not only distinguish the absorption lines of methane, but the different isotopes of methane. It used to be that measuring methane isotopes anywhere was a challenge, much less in the field, but as of 10 or 15 years ago, we can now do this with high precision in the field, and we can couple this with automatic chambers. Next slide, which when we flow gas through those chambers and into the system, we get the next slide, which shows how we can get really high resolution data of methane across this thaw sequence. The brown points indicate the intact permafrost, the green points the partially thawed permafrost, and the blue points the fully thawed permafrost, which we can see there's a big increase in methane production. But critically, we can also get the isotopic composition of these methane fluxes, which indicates the metabolic pathway by which the methane was produced. And this is our link. This is the connection to the microbial scale, because it's those microbes that are doing those metabolisms. So if we see the next slide, we can collapse that whole time series of data from 1 year into a single point shown on the upper panel here, which shows the time sequence within each thaw stage, pulsa, bog, and fen, and across multiple years indicated by each point, where each point is the average of a whole sequence of like what I just showed you. And you can see the general rise in methane emission as we move across the thaw sequence. Next slide shows the critical addition to this, which is the methane isotopes. And so we can see we're real-time measuring these methane isotopes in the field. And you can see a distinct shift towards heavier isotopes from the green thawing, partially thawed region to the fully thawed blue area. And this is a shift from hydroneutrophic to acetoclastic metabolisms of methane production. And if this is really happening, as we can see at the ecosystem scale, we should be able to match it up with what the microbes are doing at the microbe scale. So go to the next slide and we can see whether that's happening, and indeed it is, because we can see we've shifted the microbial community composition from across the thaw stage, and that we're detecting this with just a very simple technology from genome sequencing, looking at the relative abundance of this RNA ribosome gene, which is a critical gene for all of life, and so it's in all organisms. So we can use it as a kind of name tag to see who's there. And what we see here is a shift from the green organisms, which do hydroneutrophic methanogenesis, to the purplish organisms, which do acetoclastic. So we see, next slide, a key initial result of this work was concordant responses of isotope geochemistry on the one hand and microbial communities on the other, which is illustrating the possibility of scale. So now with these two technologies, we can see the shoulders of the giant we stand on, next slide, which are Joseph von Fraunhofer on the left, we could go to the next slide, and who was an 18th century, I'm sorry, we're getting hit. Let me back up a second. So who are these microbes in these pie charts? So it turns out that most of the green is a single organism, a hydroneutrophic methanogen called Methanolflorens stordalomarensis. And the next slide shows that this is very highly correlated, very well predicts the methane isotope composition, or rather the effective fractionation of methane of the carbon isotopes. So next slide, this shows a connection between gene scale information and ecosystem process, and highlights the potential importance of microbial ecology for understanding global climate change. And it uses the two axes, illustrate those two technologies, molecular gene data and ecosystem biogeochemistry. And that is, of course, is the giants whose shoulders I want to emphasize. Next slide, which are on the left, Joseph von Fraunhofer, who discovered spectral absorption lines and invented the spectrometer, and basically is the technology underlying the detection of methane isotopes that I referred to. And much more recently, Carl Rose, who discovered the third domain of life, the archaea, which are very distinct from bacteria, a completely different life form. And the archaea include the methanogens, which we've been studying. And incidentally, he also invented the phylogenetic taxonomy via using this, sequencing this 16S ribosomal RNA gene, which is the way we were identifying the relative abundance of those different organisms. So that's what we've done so far. What I want to do is show what we're doing next. Now, that was a paper in Nature 7 years ago. It's now been made obsolete. And so what we're doing now is trying to go beyond single organism correlations with methane cycling to do multidimensional community controls on organic matter cycling across the whole carbon cycle. And this is really enabled on the left. We can go beyond just looking at who's there from those 16S ribosomal genes to looking at the assembling whole genomes of all the organisms in an ecosystem. And this has really only been possible in the last 5 or 10 years, where we can actually get a representation of a significant fraction or even majority of the microbial community represented each by their genomes, which not only allows us to say who's there, but what their metabolic functions are. And then on the right, we can link this with new technologies for measuring the metabolomic data, the whole suite of thousands of elements, as you can see from the peaks of that spectrometer. That's a mass spectrometer that is now able to detect at very high resolution thousands of organic matter compounds and link those, couple them with those are the products and reactants of all the reactions that are catalyzed by those microbial communities on the left. And we're able to now do this, if you go to the next stage, next slide, a work in progress led by Malak Tufeli and Joel Boyd, which is depicting here how we can marry networks of metagenomics, that is the metabolic capacity of the ecosystem, with metatranscriptomics, which shows what's happening at the moment via gene expression, with metabolomics, which are the reactants and products that those reactions are using and producing. And what we see here on the left is the partially thawed bog, and on the right, the fully thawed fen, a network where the circles are the metabolites, the lines with the little triangles are the reactions that we discern from the metagenomic sequencing, and the heaviness of the lines, which you can see are very dark on the right, is the intensity of the transcription of those. We can see a coherent picture of a piece of the carbon cycle in action here across all three of those kinds of information, showing a coherent shift in carbon cycle biogeochemistry and acceleration and intensification, because those black areas are bigger as we move across the thaw gradient. So that's where we are now with this challenge of scaling from genes to ecosystems. And if we show the next slide, we are now in a position, well, this is sort of a summary of what I just showed you. First, empirical demonstration of genes to ecosystem scale linkage with these new technologies. And secondly, that's an empirical correlation, but no mechanism. But now we can look at the second part of the lower part of the slide shows mechanisms that link genome scale information to ecosystem processes. So now we can do this in the field. Let's see how we can test these relationships more rigorously on the next slide, which brings us to Biosphere 2. So this is really a test bed for testing molecule to ecosystem scaling in controllable environmental settings. And what I'm going to focus on is the central area that you can see here, which is the Landscape Evolution Observatory at Biosphere 2. And what this is a laboratory for looking at ecosystem scaling. And the things we need to the technology we need to test our scaling theories are three things. Number one, it has to have scale, which means has to be measured at different scales. And three, finally, it has to be controllable and manipulable so we can see the effects of precisely created different conditions. So next slide shows the Landscape Evolution Observatory, and we can see it's 3 different hill slopes that are 30 by 10 meters. And the next slide shows that this is of a large scale. You can see that this is a big system. And so it meets that criteria. Its next slide shows it's measurable, like no real ecosystem is. We have 1,200 sensors per slope. This is showing just different layers in the soil that are measuring gas concentrations and temperature and moisture and water content and all these things. And finally, the next slide shows that we're also measuring this in the atmosphere above the Leo hill slopes, getting at temperature, wind speed, and gas fluxes, things like carbon dioxide and methane and water. And we're looking at this next slide with remote sensing tools, so we can see very high precision what the landscape is like across this scale. And finally, the next slide, we're also looking at the runoff of this hill slope, which is caught by 6 outflow sections with tipping bucket gauges, and which is capturing the microbes that are in this hill slope, which are running off with water, as well as the geochemistry. So then we can apply those same technologies that I was just talking about for thawing permafrost in Arctic Sweden here within the Leo hill slope. So this brings it all together because it really gives us, next slide, the basis for investigating hydrological, geochemical, biological interactions at multiple scales, all the way from those gene scale organisms, which we can get from sequencing the genes we find in this hill slope, up to the fluxes that we are able to capture because this is an enclosed system, and then the experiments we can do because we can manipulate this as we watch this landscape evolve over time. You
have 1 more minute, Scott. Just want to put it there.
Okay, well that pretty much brings me to the end of what I want to say. So the last slide actually just acknowledges our funding sources, which include DOE and the many institutions that are involved with us. And so we're now really poised at the exciting stage of Leo, which is to actually put, you saw that those photos I showed just have bare soil, and we're now at the cusp of, and we've been using that to investigate the geochemistry of weathering and the hydrology of how water moves through that landscape. And now what we're really going to be able to see is how when we put life on Leo, we put plants. Of course, there are microbes there already, but we're now on the cusp of adding plants. We'll really be able to use these technologies that I described within the technology itself of Leo to investigate these processes at all different scales and bring them all together.